Greetings, everyone, and welcome to another Genetic Engineering and Biotechnology News webinar. Our presentation today is entitled Accelerating Ion Channel Characterization and New Drug Candidate Identification. I'm Jeff Bogaliskas, Technical Editor for GEN, and I'll be the moderator for today's webinar presentation. The ever-increasing pressure to accelerate drug development while maintaining high data quality has resulted in advancement of enabling technology. In the world of ion channel-focused drug discovery, this has involved the development and evolution of high-throughput electrophysiology platforms that maintain the high fidelity of the gold standard patch clamp platform while dramatically increasing throughput. In this GEM webinar, we'll focus on high-throughput functional annotation of human ion channel variants associated with excitation disorders. Additionally, we will discuss the automated electrophysiology platform, the Syncropatch 384PE, and its ability to enable a better understanding of how human disease-associated mutations alter ion channel function, as well as facilitate drug candidate characterization. Let's meet our panel, who will describe current research methodologies that incorporate the use of the Syncropatch 384PE for measuring subtype selective activation of Kv7 potassium channels, as well as the inhibition of various voltage-gated sodium channels. Our first speaker today is Alfred George, Chairman in the Department of Pharmacology at Northwestern University Feinberg School of Medicine. Dr. George will describe how he has utilized the automated patch clamp system to characterize an array of ion channel variants. Our second speaker will be Matt Fuller, Senior Scientist at Icogen. Dr. Fuller will outline how the Syncropatch 384PE can be utilized not only for ion channel disease characterization, but also for the discovery of novel drug therapeutics. So before our speakers get started, I want to encourage the audience to submit questions for our Q&A session at the end of the presentation. Our speakers will be on hand to answer as many questions as we can. Just simply type the question into the Q&A box on the left-hand side of your screen and hit submit. So, without any further ado, let's get our webinar underway. Al, the floor is yours.
Thank you, Jeff. My talk today will focus on our use of automated patch clamp recording to do functional annotation of human ion channel variants. This is an opportunity for our lab to make a contribution in understanding channelopathies. Those are genetic disorders involving mutations in genes that code for ion channels or ion channel regulatory proteins. And over the last 20 years or so, there's been an emerging and massive tsunami of genetic variants discovered in individuals with a variety of inherited conditions such as epilepsy, cardiac arrhythmias, hypertension, many other diseases. And the ability to keep up with studying those variants has really been eclipsed because of the large number of mutations identified. For example, on this slide lists a number of genetic arrhythmia syndromes that have been identified in association with mutations in either pore-forming or accessory subunits to various cardiac ion channels. The first disease listed, the congenital long QT syndrome, is the one I'll focus on today. And that's a condition that's been associated now with mutations in 16 different genes. Many of them are themselves ion channels or some kind of regulatory protein. The most commonly mutated gene, KCNQ1, is responsible for about 45% of all known cases of long QT syndrome. And most of the mutations that have been published are cataloged very nicely by the Human Gene Mutation Database. And so I went into that just to produce this table to illustrate the scope of this problem of genetic variants in ion channels. This lists the number of known mutations or genetic variants associated with congenital long QT in KCNQ1 and a few of the other major genes. And of note, again, relevance to this talk is KCNQ1 is associated with well over 500 mutations that are known in the literature. Importantly, only a small fraction of those have been subjected to rigorous functional experimentation to understand what their major abnormality is. And nowadays, with widespread use of genetic and genomic testing in patients with these conditions, there is a growing concern that many variants can't be properly classified as to whether they are pathogenic or benign. And so I'll make the pitch that we need more functional annotation of genetic variants to be able to solve that problem. This is a very challenging topic, and in the conventional approach to understanding the functional consequence of genetic variants in any ion channel, including KCNQ1, which I'll talk about today, the traditional approach has been to use manual patch clamp recording, using heterologous expression of a human recombinant ion channel cDNA in a non-cardiac cell, for example, in long QT syndrome, with the goal of being able to study individual mutations over the course of several days to a few weeks. And this is basically because of the limitation of conventional patch clamp recording, even the best, most experienced patch clampers, like the three featured in this slide, these are members of my laboratory, at best they can probably get on average maybe 16 good measurements a day, and therefore it takes several days to maybe a few weeks to do a thorough job of studying an individual mutation. And so when faced with the tsunami of genetic variants, this method is really not up to the task. So 2 years ago, we installed the first automated patch clamp instrumentation at Northwestern University, and this was the Nanion Syncropatch 768 patch engine, which I'll just call the Syncropatch. This is a dual 384-well planar patch clamp system that's integrated into an industry standard liquid handler, Biomech FX robot. And this is really capable of doing a lot more work than conventional patch clamp recording. There are several features of this instrument that convinced us this was the platform to go with. Without sounding like an infomercial, I just want to list those features here. We thought it was very important to be able to have the capability of having gigaohm seal resistances in the planar patch system, because of the high fidelity of the voltage clamp and high quality of the recordings that we're used to with manual patch clamp recording. There are several other features that are listed here. We were very attractive with the ability to have two 384-well plates simultaneously running. There is now an opportunity in the near future to have current clamp in addition to voltage clamp recording. And the fact that this was integrated into a liquid handling robot, I think that's an industry standard, was also very attractive to us. There was another challenge that we faced, which was normally automated patch clamp platforms work well with stable cell lines, and we were interested in studying hundreds of genetic variants. And that created an opportunity to come up with a scheme to get around the need for stable cell lines. And fortunately, a woman I hired who formerly worked for a big pharma suggested we use an electroporation platform made by the Maxite company. And so featured in this slide is really the tandem combination of these two technologies that really allowed us to move forward in really kind of a quantum leap over what we were doing before. So rather than making stable cell lines on all of these genetic variants, we use transient expression with the Maxite electroporation system, and that has given us enough efficiency and high-level expression to go directly into automated patch clamp recording. The channel I want to focus attention on now is KCNQ1, which is the pore-forming subunit of a potassium channel complex responsible for the slowed delayed rectifier current in the heart, known as IKS. Now, IKS depends on the assembly of casein Q1 with its accessory subunit casein E1 that's featured here. And both of these genes, the reason they're next to chromosomal ideographs is because both of these genes are known to be associated with mutations causing congenital long QT syndrome and some other types of inherited arrhythmias. So we were interested in studying mutations in both of these genes. But today I'm going to mainly focus on casein Q1. There were 3 experimental paradigms that we had tested out on a panel of 50 genetic variants that we created in the laboratory for this project. First, that we studied transient expression of the pore-forming subunit by itself. We secondly introduced casein E1 first in a stable cell line, a CHO cell line that stably expressed casein E1. And then on top of that, we did transient expression of casein Q1. And then later, we optimized conditions that would allow us to co-transiently transfect casein Q1 and casein E1 together. And I'll walk you through the logic behind doing that. It turned out that the stable cell lines that expressed casein E1 alone weren't as stable as we wanted them to be. After several passages, the expression seemed to diminish. And so we gave up on that idea with the idea of just optimizing transient expression of both casein Q1 and casein E1, which I may slip up every once in a while and just call Q1 and E1, so just so everyone's aware of that. The experimental workflow is illustrated on this slide, which is we would use the MaxCyte system to electroporate CHO cells with both casein Q1 and casein E1 cDNAs that were built to have different co-expressed fluorescent proteins. I'll illustrate that a little better on the next slide. After electroporation, the cells recover briefly, and then you plate them in a flask to then grow for 48 hours at 37 degrees. At that point, we could go directly to patch clamp recording, but instead, we actually cryopreserve the cells so that we can then bring out batches of cells later in a queue to maximize the throughput on the automated patch clamp. And this intermediate step of cryopreservation only reduces the number of viable cells very minimally, and so we've been very happy with this as a step in the experimental workflow. The day before we're planning automated recordings, we actually thaw and replate the cells in a smaller dish, grow the cells briefly overnight at 28 degrees, harvest the cells, and then plate them for both patch clamp recording and also quality control. We do flow cytometry to estimate the proportion of cells that were transfected. So this slide gives a little better idea of what we're doing. The 2 plasmid renderings on the left show you the design of the 2 plasmids. Casein Q1 is coupled to expression of GFP through an IRES vector configuration, whereas casein E1 is coupled to expression of a red fluorescent protein through a similar vector. And this allows us to do flow cytometry and quantification of the percentage of cells that have been successfully transfected and are expressing these 2 plasmids. So what's illustrated in the flowgrams is in the left panel is both green and red, but what we've quantified is the proportion of cells that express GFP, and in this case it was 94.3%. And then on the right panel is we quantify the proportion of cells that express the red fluorescent protein. In that case it's a little bit under 83%. We estimate viability using a Beckman Vicell automated cell counter, and the percentage of co-transfected cells, which we infer from the flow cytometry data, was around 80%. And when we apply those cells to the Syncropatch, we see a very high proportion of the wells in the 384 well planar patch plates that express an outward, slowly activating current resembling IKS. And in this experiment, it's probably hard to see on the slide, but there was approximately 88% of the wells had a gigaohm seal, a cell capture, a gigaohm seal, and most of those had a slow delayed rectifier. Now there are some wells you can notice, so we've cherry-picked some of the wells in the big plate view to highlight currents on the right, and some of them clearly are not an IKS-like current. And so this evoked and motivated us to employ one more step in addition to the voltage clamp protocol to try to clean up these measurements. And that extra step included a final application of an IKS-specific blocker known as HMR 1556, and we would then record again after application of that compound and then take the residual currents and digitally subtract them offline from the total current to remove any background non-IKS current. And when we did that, we were able to eliminate any non-IKS cells and improve the quality of the measurements. In the beginning, when we began to use the Syncropatch, we asked the basic question, which is how well do these recordings mirror what we were seeing with manual patch clamp. And so just to illustrate that for the IKS experiments, what I illustrate in this slide on the left is an average of about 16 manual patch clamp recordings from cells, CHO cells, that express casein Q1 and casein E1. And there again is the familiar slow delayed rectifier profile that is familiar to those that study IKS. On the right is an average of many cells, I don't remember the number, but probably over 100 cells that were recorded by the Syncropatch. And also we see this slow delayed rectifier gating pattern, but what should be obvious is that the current amplitude is lower. Now on average, we think that's the case because when we do manual patch clamp, there is a bias toward visually identifying and then recording from those cells that have the brightest fluorescence. Whereas in the Syncropatch, it's blind. We only select cells because they were captured and from which we were able to record. If we normalize these current recordings to their peak, we can see that the gating behavior is probably identical. As identical as one can tell by eye. And here we have current voltage relationships that demonstrate a similar phenomenon. On the left, again, it's the unnormalized current showing manual patch recordings for 16 or 18 cells and Syncropatch recordings for 140 or so cells. And again, there is this bias toward being able to identify cells that have a higher current density by manual patch clamp than the automated patch clamp. But if we normalize, and that's the IV plot on the right, we see that they superimpose very nicely. And in fact, Boltzmann fit to calculate the V1/2 for activation in this case is not significantly different. And so just one more screenshot of an actual experiment where we studied in this case, 4 different variants plus wild type and non-transfected cells, just to show some real live data here with the diversity of expression. Here we actually biased the experiment to show variants that we knew had robust current and wouldn't just give us a dud. But you can also see in the far right side of the 384 well screen display, the non-transfected cells have virtually no current, and on the far left are the cells that have wild type. And this screen display does not normalize the current amplitude, and so everything looks a little bit heterogeneous, but this is a more real live experimental result. And also in this experiment, I think there was over 90% of the wells had cell capture and a gigaohm seal, so a very high output day. Now we've been able to analyze data from these experiments and have been working on automating some of the data analysis routines because it's a large amount of data to handle in the manner that one would do with conventional patch clamp data sets. And just to give a snapshot, these are some early analysis schemes that we've applied to some of the data, and we can easily average current traces up in the upper left-hand corner there. We can look at activation and deactivation time constants. We can plot current voltage and activation voltage dependence. And so these are things that we normally would do with manual patch clamp data, and we've not had any major limitations so far with KCNQ1 in terms of the data analysis side, other than handling the large volume of data, which we said we're continuing to develop automated routines to make that work better. Now, just briefly, I promised I would just veer off from the KCNQ1 just briefly to talk about some other studies that we've been doing with sodium channels. And I know that Matt Fuller from Icogen, the next speaker, is going to talk more about this as well. But we've also have felt the pressure and the need to automate functional annotation of sodium channel variants that have been identified in patients with a variety of human conditions. And this slide simply lists the voltage-dependent sodium channels on the left. Across from each of these different ion channels are the conditions that have been associated with those. And so many of these are associated with epilepsy or cardiac arrhythmias or a variety of peripheral nerve problems. We've been mainly interested in the epilepsies and cardiac arrhythmias, and so I just want to just touch on some data that we've been doing, again, around the idea of doing high-throughput functional annotation of genetic variants in these diseases. This slide illustrates some of the work we did early on, right after we got the Syncropatch installed, looking at expression of NAV1.1. This is a very early experiment. We actually used stable cell lines in this case, and we're very happy with the early success of the instrument. Again, we've compared manual patch data with Syncropatch data for NAV1.1, and as you can see in this slide, the voltage dependence of activation is almost perfectly superimposed, whereas steady-state inactivation is a little shifted for the Syncropatch, and there is a calcium-rich solution that's applied as one is achieving the initial to enhance the initial seal resistance. And we have found it necessary to do some extra washes to remove that excess calcium. We think that contributes to this slight shift in the steady-state inactivation. And then in a follow-up experiment, we looked at stable expression of the wild-type channel and a mutant that we had studied many years ago that's associated with a genetic form of epilepsy called GEFS+. And this is a mutation in the domain for S4 voltage sensor segment that is associated with more or less normal gating behavior, but an increase in late, or also known as persistent sodium current. And we wondered if we were able to see persistent sodium current using the automated patch clamp system. And this slide illustrates that we were able to see it. Here on the left is an average of wild-type and mutant recordings that are expanded out to 200 milliseconds, the duration of the depolarization in this experiment, and normalized to the peak current. And this illustrates very nicely a level of around 2.5% to 3% persistent current that mirrors very nicely the manual patch clamp experiments that we did. We are also interested in using this approach to study variants of unknown significance that have been identified in patients with a different form of genetic epilepsy called Dravet Syndrome, a very severe epileptic encephalopathy for which there are now more than 1,200 known mutations in NAV1.1, which is encoded by the gene SCN1A highlighted on this slide. And so we've actually done some work to look at the capabilities of the Syncropatch to do functional annotation of genetic variants in this condition. And what's illustrated here are some average recordings from one experiment where we looked at 4 different variants associated with Dravet syndrome, 3 of which are completely non-functional, And that's a result consistent with what's known about this disease being a loss of function of NAV1.1. But the fourth mutation really has an intermediate phenotype. It looks like it has pretty normal gating behavior, but it does not have the same current amplitude as a wild-type channel, but it's not a complete loss of function. So this is something that we think is important to document for the large number of variants that have been associated with this condition. And then really lastly, I just have a couple more slides. The first, as shown here, is our initial foray into looking at cardiac sodium channel variants. This is a slide that depicts 3 different variants. I've only labeled wild type and one of them just to highlight in this talk. But this is the cardiac sodium channel that we've transiently transfected using the electroporation system from MaxCyte into HEC293 cells. And again, on the right are some summary or average current traces for both wild type and this particular mutation, G1631D, which was associated with a very malignant cardiac arrhythmia in young children. And in fact, by manual patch clamp, we were able to see this very slow inactivation time course and a large late current component, and it's really indistinguishable from what we were able to pick up in this Syncropatch recording. And so just to sort of summarize a little bit and show you one little last tantalizing tidbit of data, which we published recently, our long-term goal is to use the high-throughput automated patch clamp system to do functional annotation of genetic variants, and that's what I've been illustrating. But then to go beyond that at some point as a kind of a paradigm of precision medicine and be able to screen compounds, both approved drugs and maybe some investigational compounds, against selected variants that have particular functional abnormalities, with the goal being is to create a pharmacological profile for an individual variant, and that may in some situations help identify the right drug for the right patient that has either an inherited cardiac arrhythmia or an inherited epilepsy. And as kind of a proof of concept, a couple of months ago we published a study that was largely done by manual patch clamp recording to look at the biophysical properties of a novel sodium channel blocker known as GS967, and that is a selective late sodium current blocker, although we identified that it can also cause a use-dependent block of the peak current. And we wanted to compare the concentration response of GS967 against a couple of other sodium channel blocking drugs and do that in an NAV1.5 stable cell line. And what's illustrated here are work from our molecular pharmacology publication from earlier this year, which illustrates the power of this automated patch clamp system, where we were able to perform concentration responses with 7 concentrations for 3 different drugs at different frequencies. And these experiments required 2 384-well plates that were done on 2 different days in 1 week. So this is an amount of data that would not have been possible to collect by manual patch clamp without an army of people and a lot more time. And so we were really pleasantly pleased, and this is a nice transitional slide to the next talk, which is going to really talk about how this platform is used for drug discovery and other pharmacological applications. I'll last leave you with this acknowledgement slide of the people in my group who contributed to this presentation. I want to highlight Carlos Vanoye, who is second from the left in the back. He's the one that really has been the champion of operating the Syncropatch in our group. I also highlight the work of Reshma Desai and Katerina Fabre, who are the second and third in the front row from the left, and they've done a lot of the molecular biology and tissue culture for this study. And a lot of the work I've shown you today is funded by a grant from the National Heart, Lung, and Blood Institute. Thank you for your attention.
Thanks, Al. That was an excellent presentation. I think you gave the audience tremendous insight into the use of the Syncropatch 384PE for characterizing human channelopathy. So before we move on to Matt's presentation, I want to remind everyone once again, submit questions for our Q&A section at the end of the presentation. Just simply type your question into the Q&A box on the left-hand side of your screen and hit submit. Both of our presenters will be on hand to answer all of your questions after the final presentation. All right, with that said, let's move on to our final presentation. Matt, the audience is listening.
Thank you, Jeff, for the kind introduction. I'm with the Acknowledgements team, and our organization is a contract research organization, so we're probably using the Syncropatch system in a slightly different method than what Dr. George's group is using it at Northwestern. So we're more involved in the second part of this title, the new drug candidate identification. And at our site in North Carolina, we have a broad technological capabilities that allow us that support small molecule and biologic drug discovery, but we also have custom tools such as custom cell lines that may enable us to identify unique subtype selective ion channel active compounds. But we also have stem cell based platforms that can allow for translational studies beyond heterologous expression systems. But today's talk is more focused on our most recent technological acquisition, which is the Nanion Syncropatch, which is shown here on the top left. And what I'll talk about today is our two recent assays that we have developed for screening compounds against two clinically relevant targets. The first target is the voltagegated calcium channel KCNQ. Al had just spoken about KCNQ1. We will be focused more on the neuronal KCNQ channels Q23 and Q35. And for this presentation describing the Syncropatch assay, we'll first talk about some linked channelopathies and how they're related to KCNQ. We'll discuss why people are interested in identifying KCNQ openers and what the mechanism of action is behind an opener. We will also discuss and show examples of KCNQ 2.3 and 3.5 recordings from the Syncropatch. We'll talk about control, compound data reproducibility. And we will also do some more detailed studies looking at channel subtype selectivity, as well as some binding site identification for known KCNQ openers. So KCNQ channels are known to be expressed in neuronal cells, especially KCNQ 2.3 and 3.5, while KCNQ4 is expressed in auditory hair cells. And loss of function mutations in any of these channel types can cause hyperexcitability disorders, ranging from benign familial neonatal convulsions all the way up to generalized clonic seizures and epilepsy. So you can see that because loss of function mutations cause hyperexcitability disorders, there's a market there to identify openers that could make up for some of the loss of function that occurs due to these mutations. And currently there are KCNQ openers on the market, one of which is Reticle Beam. So this compound is effective at opening Q23 as well as KCNQ35 channels. And here, this slide is just giving you some information of how a KCNQ opener works, what the mechanism of action is. So these are conventional patch recordings of human KCNQ2.3 expressed in a CHO cell line. So when we record these currents, we record full IVs across a broad range of membrane depolarizations in both control and with the opener present. And as you can see, when in the portion where there's 3 micromolar Reticle Beam applied to the cell, the tail currents are much larger. And we also can see that it appears as though the channel is activated at more hyperpolarized potentials. To determine that this is true, we measured the peak tail currents from following each depolarization. We then convert those currents into conductances, and we plot normalized conductance versus voltage. We then fit those points using Boltzmann equations to give us an activation curve of the Q2-3 channel in both control conditions and with ascending concentrations of Reticle Beam present. And as you can see, as we go higher in concentration of the opener, we get a more and more hyperpolarizing shift in the activation curve of the channel, suggesting that indeed the channel does become activated at more hyperpolarized potentials when the opener is present. From this data, we can determine that voltage required for activation of 50% of the total current. This V-half for activation is shifted along with the curves. So by determining what the shift in V-half is, we can then plot that, which is shown on the far right on the top, as plotted as the delta V-half for activation versus the concentration of the compound applied. We can then fit those values to give us the maximum response obtained from that compound, which in this case is Reticle Beam, and we can also get a measure of the EC50 of this compound, which for us in conventional patch recordings is 5.7 micromolar. So this type of opener shifts the gating of the channel, allowing it to open at more hyperpolarized potentials. So how does this reduce neuronal excitability? Well, to answer that, we performed current clamp recordings of primary human dorsal root ganglion neurons in both control conditions and with a second KCNQ opener, ICA 105665. Under control conditions, the channel opens at potentials near the threshold for action potential firing, and you can see that there's numerous spontaneous action potentials in the control recording. However, when the opener is present, the channel opens at potentials closer to the resting potential of the neuron. This causes a hyperpolarizing shift in the membrane potential of the cell, which makes it more difficult to reach the threshold for action potential firing, thereby reducing the number of action potentials we observe per sweep. So basically it's an overall depression of neuronal excitability, which is what you would want to see if you were trying to identify therapeutics for hyperexcitability disorders. So when we were developing the assay on the Syncropatch, we wanted to have it mimic what we had done in conventional patch recordings. We wanted to run full IVs, and if we could, we wanted to do this. We wanted to run the two channels side by side on the same chip. So an example of which is shown here. So these are full IV recordings of both Q23 and Q35 on the same chip. These are 9-hole population chips where we routinely get 84% of the wells giving us usable data for every recording. On the right is example or larger pictures of individual KCNQ35 recordings, both in controlled conditions as well as with 30 micromolar Reticle Beam. And as you can see in these recordings, the tail currents again are larger than what they were in the presence of Reticle Beam than versus control, which suggests that the compound may be acting in the same way on the Syncropatch as it does in conventional patch mode. To determine if that was true, we did the same analysis as we had done with our conventional patch recordings, and when we plotted out the delta V-half for activation versus the concentration of Reticle Beam applied to the cells, we find that Reticle Beam is equally effective against both 2,3 and 3,5, and that the magnitude of the effect is equal to that had been observed in conventional patch recordings. And in the table below, you can see the parameters that we observed from these experiments as compared to the conventional patch recordings. And the EC50s are right in line with what we had seen before from conventional patch. The two bar charts on the top, the one in blue is for KCNQ35, the one in green is for KCNQ23. The colored bars are in the presence of Reticle Beam at 10 micromolar. The gray bars are DMSO. And what we're showing here is that over repeated applications or repeated chips covering 3 different days and 6 different chips, we get reproducible data, suggesting that the assay works perfectly fine, it's reproducible, the compound behaves as it should, and the Syncropatch assay matches very well to what we have seen previously with the conventional patch recordings. To follow that up, we also tested 2 other previously published KCNQ openers, ML213 and ICA 69673. Each of these openers behave similarly to what they had done in the published work. The ML213, the EC50 is almost exactly what it is off of the Ironworks Quattro in the U paper from 2011. And the 69673 compound, the EC50 compares favorably to the rubidium flux assay results that were also previously published, suggesting that this assay works perfectly well for identifying KCNQ openers if you're looking for the amount of shift in the activation curve of when those openers are present. But as I was saying before, we wanted to be able to run both channel types on the same chip because we're also looking for subtype selective KCNQ openers. So previously, the group here at Icogen has identified compounds that selectively open Q23 but not Q35, and one of those examples is shown here, ICA 27243. And underneath are normalized conductance versus voltage curves for both channel types in the absence and presence of either ICA 27243 or Reticle Beam. And as you can see, Reticle Beam works equally well on both channel types as we have observed from the Syncropatch recordings. However, 27243 is only effective when applied to the Q23 channel. There is no effect when it was applied to the 3.5 channel. So we wanted to know if we would get similar observations if we applied the same compound using our Syncropatch assay. So here is the results from applying ICA 27243 to either Q23 or Q35 on the same chip during the same experiment. And what we find is that the EC50 for activation of Q23 is similar between the Syncropatch and the conventional patch recordings shown in black in the center figure. There's less than a 2-fold difference, so essentially this is the same result. And importantly, if you look at the Q35 results, again, 27243 failed to activate the current, which suggests that the KCNQ subtype selectivity is consistent across these 2 platforms. So the Syncropatch is giving us similar results to what we had seen previously from conventional patch once again. Finally, we wanted to look at some mutant forms of the KCNQ23 channel and see if the change in pharmacology we had previously seen matched up with the Syncropatch. So the Reticle Beam binding site has been identified for a while. We have a mutant form of KCNQ23 that has 2 residues mutated. That when we tested them in flux, we found that Reticle Beam was not active against the mutant form of the channel, but activated the wild-type KCNQ23 just fine. So that is shown on the top as a % activation versus concentration of the compound. So Reticle Beam is affected greatly by this mutation. However, when we repeated the same experiments using ICA 27243, we found that compound was not affected by the mutations, suggesting that it had a second binding site away from the previously identified Reticle Beam binding site. And more recently, a paper has been published illustrating this effect. So the structure on the left illustrates where the Reticle Beam binding site is in the pore region of the channel, and the new residue, the 2 identified residues that are shown to affect ICA compound binding, which are both present in the voltage sensing domain of the channel. So we wanted to determine if we could get similar results on the Syncropatch as what had previously been shown in the flux assay. So we used CHO cells expressing the mutant form of the channel or the wild-type channel. We applied those to the Syncropatch at the same time on the same chip, side-by-side recordings, and then we tested either Reticle Beam, ICA 27243, ML213, or ICA 69673 against both channels. And what we find is that for the mutant channel, ML213 and Reticle Beam had no effect on activation. However, the ICA compounds behaved just like they had done before with the wild-type channel, again suggesting that if you have compounds that interact with the channel at sites away from the common Reticle Beam binding site, the Syncropatch can effectively identify those compounds as openers and show those positive results. So all of the data from the KCNQ assay is consistent with previous findings, suggesting that this assay is adequate for identifying selective KCNQ openers. The second assay that I will describe is voltage-gated sodium channel assay that we've developed on the Syncropatch. I'll show you examples of NAV11 and NAV15 recordings, and then we'll show some data describing subtype selectivity as well as state dependence of block using the Syncropatch system. We'll also talk about newer generation of voltage-gated sodium channel inhibitors that interact with the voltage sensing domain, and we'll describe our results when using those compounds on the Syncropatch as well. So as Al mentioned, voltage-gated sodium channels are expressed in numerous tissues, and some mutations can result in disorders such as epilepsy. Here we're showing 3 different NAV channel subtypes that are expressed in neurons, NAV17, 18, and 19. Each of these, the mutations that are depicted here, have been associated with modulation of pain, which makes them a good target for therapeutics for the treatment of pain. So when we developed this assay, we also wanted to have the ability to run 2 different cell lines at the same time on the same chip for rapid evaluation of compound selectivity, but we also developed the voltage protocol to allow us to look at resting state block and inactivated state block while we were also looking for subtype selective compounds. So this is a highly flexible assay in our hands, and I'll show you examples of that in future slides. So here we're looking at subtype selective inhibition of NAV11 or NAV15. So these were recordings done simultaneously side by side on the same Syncropatch chip. So these recordings, I should have said previously, these recordings were done on single hole chips. So it's 1 cell landing per hole instead of previously when there were 9 cells per hole, per well that were landing for the KCNQ experiments. And again, we usually get 75% of our wells that give us usable data per experiment. So it's a highly efficient assay to run. And what we're looking at here is tetracaine and tritilotroc and TTX block of either 1,1 or 1,5. Tetracaine blocks both channels equally well when applied to the inactivated state of the channel. As you can see, the IC50s are essentially the same. For TTX, however, it's more efficient at inhibiting NAV11 than it is NAV15, which is consistent with previously published results. When we get the state dependence of inhibition using these 2 compounds against these 2 channels, we also see consistent results. So when tetracaine is applied to NAV15, it's potent against the NAV15 inactivated channel with an IC50 of 1.7 micromolar. However, it doesn't really block the resting channel at all. However, TTX, when applied to NAV11, shows no state dependence at all. But all of these findings are consistent with previous results, as these are well-behaved and well-understood compounds. However, now we want to transition over to the newer generation of NaV1.7 inhibitors that have been developed by Pfizer. These compounds have previously been shown to interact not with the pore domain as TTX or local anesthetics, but rather in the voltage sensing domain of domain 4 of the channel. These compounds are very potent, however, they are very state dependent as well. So the graph on the bottom right in black shows application of 100 nanomolar of PF771 to the resting state of the channel, and essentially there's no block. However, when this compound is applied to the inactivated state of the channel, you get complete block. But there's also another caveat to these compounds. Some of them block very slowly. As you can see, yes, 100 nanomolar 771 results in complete block, but it takes 15 minutes to get there. As you use lower concentration of the compound, it takes longer and longer to reach steady state inhibition, which combined with the significant state-dependent interaction of these compounds make them very difficult to work with when using planar systems to identify them. So we wanted to test each of these compounds on the Syncropatch to see how they matched up with our previous findings. So for PF264, we tested the compound on the Syncropatch and compared that to what we had seen in the conventional patch. And indeed, the IC50s are almost the same. The compound behaves very well on the Syncropatch. For PF771, we see the same thing. When we compare the Syncropatch results to those previously obtained from the Patch Express, we see the IC50s are almost the same, suggesting that even though these compounds are difficult to work with, and they do have novel binding sites away from the common inhibitor binding sites of other sodium channel blockers, the Syncropatch works very well in identifying these compounds and finding their true potencies. So, as a summary to this, we find that for KCNQ and Voltagegated sodium channel pharmacology on the Syncropatch, the system is very efficient. The Syncropatch provides hundreds of high-quality recordings on each chip. It's accurate. The potency and efficacy of the compounds for both KCNQ and NAV channels are in line with published results, and the results are reproducible, as channel subtype selectivity for both channel types was also in agreement with previous results. So this system appears to work very well for identifying either openers for potassium channels or inhibitors of sodium channels, even if the compounds that you're looking for may bind in novel sites or have novel modes of action. So thank you for your attendance, and Al and I would be glad to answer any questions that you have. Thank you.
That was great. Thanks very much, Matt. I think the audience now has an appreciation for the power and versatility of the Syncropatch 384PE during the drug discovery process. Looks like we've got some really great questions already coming in, but I'm encouraging the audience to keep them coming. This will be your final chance to submit questions for our speakers. Simply type your question into the Q&A box on the left-hand side of your screen and hit submit. All right, so let's start the Q&A. Just give us a moment to get everything squared away on our end, and we will start the Q&A session.
All right, everyone, so let's get to the questions. We've had a lot of them come in, some really great questions. So the first one is going to be for Al. Al, one of our audience members would like to know, can you confirm if the Synchro 768 has two simultaneous readings of the 384 well plate?
No, that's a very appropriate question. Sorry I didn't address that in my talk. So we use a dual 384 well system, and in fact we can run both plates at the same time. They are the robotics adds liquids, solutions, and cells in a staggered fashion to each plate. There's only one 384 well head, so we can only add stuff to one plate at a time. But once everything is ready to go, the two plates can operate together at the same time. Each patch engine that contains one 384 well plate is run by its own computer. And so we can actually set up completely different experiments if we wanted to on two independent 384-well plates, and they do run together at the same time. Now, whether the pulses are exactly on top of each other, I don't know that, but we can run two simultaneous experiments on two 384-well plates.
All right, thank you, Al. Matt, we have another question, or we have a question for you. First question is, are there plans to provide additional human species orthologs for NAV channels?
Yes, there is. We have the experiments I showed here were stably expressed channels in HEC-based cell lines. We have a number of human sodium channels expressed in that way, and we're working our way through each one of those on the Syncropatch. It seems like each cell line has its own particular quirks that it likes, so you have to kind of tweak your system according to which cell line that you're working with. So some work better under certain conditions, others work better under slightly different conditions. And we're working through that as we go along. We're still in the development stage with this system. We haven't had it that long, but our plans are to put any sodium channel that we have in stock on this system and screen against that.
All righty, thank you, Matt. Al, another question for you. What has been your success rate of using transient expression of NAV 1.5 or other sodium channels?
So in the examples I showed, those were, in fact, transient transfection of, I actually showed some stable and some transient. We haven't done as much optimization on sodium channels as much as we've done on the KCNQ1 project that I mainly highlighted. But I would say the NAV 1.5, we're operating probably between 65 and 75% of wells have a cell capture and have a gigaohm seal and generate pretty high quality data. So that I think we can improve on. For some of the neuronal sodium channels, so for example, NAV 1.1, which was some data that I showed, we are lower than that, and we're not entirely sure why, but we also have not spent a lot of time doing that. I want to also point out that we do the KCNQ1 experiments exclusively in CHO cells, and sodium channel experiments we do exclusively in HEC cells. And I think there are different optimization parameters for both. And we just haven't spent as much time optimizing around HEC cells, but I'm pretty certain we can do better than 75%, which is where we are right now with NAV 1.5.
All righty, thank you, Al. Matt, another question for you. Our audience member would like to know if it's possible to validate antibody drugs directed against voltage channels with Syncropatch.
That's a good question. We haven't tried that yet on the Syncropatch. We're planning on that, on testing some antibodies on there. I see no reason why it wouldn't work. Maybe we would need to tweak our system. Maybe the solution's by adding some BSA or something to ensure that the antibodies don't stick to the plastic pipette tips that we use to load the wells, or maybe to the walls of the wells once the solution has been added. But I see no reason why the antibodies would not work on this system. It's just not something that we've had a chance to get to yet, but there's definitely plans for that type of assay.
All right, thank you, Matt. Al, another question for you. Was the KCN1 stable cells a clonal isolate?
The Casein E1, oh yes, the Casein E1 stables, and I didn't really show very much data with that. I'm wondering if someone misconstrued something, but we did use a Casein E1 stable cell line for some experiments. And it was a clonal isolate. In fact, we made several clonal isolates. The best ones that we started working with, the expression seemed to peter out after only about 5 or 6 passages, and so they weren't really ideal. That's why we ultimately went to just co-expressing Casein E1 transiently with Casein Q1 transiently with the electroporation strategy, and that seemed to work much more reliably. But to answer your question properly, yes, those were clonal Casein E1 stable cell lines.
Thank you, Al. Matt, another question for you. Audience member would like to know what is your success rate on the Syncropatch for usable data?
So it varies depending on which assay that you're running. So I think I mentioned that for the sodium channel assays, I think we're averaging around 75% of the wells giving us usable data. For the KC&Q projects, those are CHO cells, which seem to work slightly better for us on the Syncropatch. And for those, on average, I think we're getting 84 to 85% of the wells that give us usable data, which is a good return, I think, for a 384 well-based system like this. So we get plenty of data points for every run when we're screening compounds. So it's a good question.
All right. Thank you, Matt. Al, another question for you. Why do you express the KCNQ1 in CHO cells and not other cell lines like HEKs?
Well, I think this is based on observations that were made over years of using manual patch clamp on these cell lines. In HEK cells, we tend to see an outward current that we think would confound our measurements of any potassium channels. So we actually just systematically don't look at potassium channels in HEK cells. And by the contrary, sodium channels, there's been some evidence that CHO cells can express an endogenous sodium channel. And again, it's not consistently observed, but we've seen it on rare occasions. And so but we never see endogenous sodium currents of any magnitude in HEK cells. Or at least it's predictable, and we know when to throw the cells out. So that's really the reason. We just don't see endogenous potassium channels in CHO cells, but we do in HEK, and that's why we favor CHO cells for that system.
All right, thank you, Al. Another question for Matt. Matt, what were the QC parameters used on the 384 PE?
So there's a number of ways that we can eliminate poor recordings or wells that just didn't have a cell that landed that gave us a seal before break-in to work with. The Syncropatch system is very good about recording everything from seal resistances to series resistance to cell capacitance, all of these different parameters that you can use to determine how healthy an individual well was or a recording was. So we use things like that. We have limits set for minimum seal resistance or minimum peak current amplitude that we use to remove cells that just didn't have the current for sodium current or a KC&Q current that we were looking at here. And because we're running full IVs for most of our assays under controlled conditions, we can also use the biophysical nature of the current itself to determine if the well the recording that we're looking at from each well is giving us the current that we're looking for. So if you run a full IV on the KC&Q current, you can look at the different parameters such as the slope of the activation curve or the potential required to give you half activation. And if those values vary one way or the other too far, then you can say, well, that's probably not the current that we're looking for. Maybe it's just a leak current that was showing up. And then we can go back and look at individual wells that have those strange results and determine just by visualization whether that was actually a KC&Q current or whether it was just a leak current that was showing up. So there's a number of ways that we can go back and determine whether we want to keep wells based off of how well the cells landed, how they sealed, and then during the recording, if we actually see current that looks like the current we're expecting to see.
All right, thank you, Matt. Al, another question for you. Why do you grow the cells at 28 degrees overnight instead of 37 degrees?
Yeah, so this question refers to a step in our current workflow. After we electroporate cells, we grow them for 2 days, then we cryopreserve them. And the reason we do that is so that we can queue up several batches of cells that we want to put on the Syncropatch all at the same time. And that helps reduce the burden of lots of acute cell culture. But when we plate the cells the day before recording, we will put them at 28 degrees overnight because of work by other investigators showing that for some of these cardiac potassium channels, KCNQ1 included, a lot of work has been done also on hERG, that there are some mutations that have reversible trafficking defects, and you can use low temperature incubation to reverse that. The reason this seems a little bit disconnected is because the follow-up experiment that we're planning to do is to not do the 28-degree incubation, to keep all cells at 37, and then compare the results, with the idea being that if there is some reversible trafficking impairment, we may see rescue of that at 28 degrees. The reason we're doing 28 first is simply strategy. If a KCNQ1 variant is non-functional even after overnight incubation at 28, we will interpret that as evidence that the cause of functional impairment is not entirely or very much related to a reversible trafficking defect. And so it just helps us streamline the big experiment. And right now we're in fact doing some follow-up on 50 variants and looking at those that had partial current or some current at after overnight incubation at 28. And the assumption is that some of those may have been rescued trafficking impaired variants. So we will likely see lower current if we do it 37. And this is just a long, this was the product of a long discussion of our pipeline and what was most efficient to do. But we do get a little boost of current amplitude even with wild-type KCNQ1, as has been observed for hERG by an overnight incubation at 28. And so it's a strategy, I think, used by a lot of people to get more current amplitude.
All righty. Thank you, Al. Matt, another question for you. Do you have any experience with population patch clamp on the fast-acting NAVs, i.e. NAV 1.7?
We have tried the population chips with the sodium channels in the past. Most of that was done when we were trying to optimize the system and the assay itself, so we haven't really tried to look extensively into that. But it does seem to us, at least in our hands with the limited studies that we've done, that it is slightly more difficult to accurately clamp the cells in that situation for the fast-acting sodium currents. But it's not. That doesn't mean that you can't do it on this system. That just means that maybe our assay was not optimized for that type of recording when we were doing that. This was several months ago when we were trying those types of experiments just to see what we could and couldn't do on this system. I think that it would probably work fine. We just haven't used it much. We haven't had to. Most of our cell lines express very well, and I think we don't really need that. You can see from the example recordings that I had in the talk, there's so many, almost all of the wells that have seals and get break-in have usable current, so we really haven't had a need to go to the population patch on the Syncropatch very much, at least not for the targets we're working with so far. But I don't see any reason why you couldn't do that. You just have to optimize the system for the plate layout and for the cells that you're using, but I think it should work. That's a good question.
Great. Thanks, Matt. And it looks like we're going to have time for one more question for Al. Al, some of the traces look somehow out of clamp. Do you have any particular strategies to improve the clamp quality for NAV channel in general, which is always difficult?
Yes, I think this question is maybe misconstruing a mutant that I showed in one of the slides. It's a NAV 1.5 mutant that, when compared to wild-type channels, has much slower inactivation than the wild-type. And that, in fact, we chose that mutant just because of that reason. We have published a paper on that mutant. It has a very abnormal inactivation kinetics. And so it does look like a channel that's not clamped well. But the big picture here is that we have certain algorithms now for analyzing data, and they all include quality metrics of the seal resistance, access resistance. For sodium channels, we look at the baseline amplitude and the peak amplitude of the recorded current to make sure that we're within a range where we should get good voltage clamp speed. And we can do series resistance compensation on the Nanion system. And so all those things taken together, we have been impressed with the quality of the voltage clamp and have not seen many problems with cells that were difficult to clamp that we couldn't throw out during the data analysis step. So I think there's not something we actively do to improve the clamp other than what is part of the routine process. But what we do is a lot of rigorous quality control on the final data so that we can eliminate those so-called bad cells.
All righty. Thank you very much, Al. And gentlemen, thank you for your answers. And with that, we've come to the end of our webinar. I'd like to remind everyone that the webinar will be archived on our site for up to a year at www.genengnews.com. So if you missed any parts of it, you can watch it again, or feel free to forward the link to your friends and colleagues. I'd like to thank Al and Matt again for their informative presentations, and I'd like to thank the audience for their attention and thoughtful questions. And a very special thanks to Icogen for sponsoring this webinar. So hopefully we'll see you again in another Gen webinar in the near future. Goodbye for now.